And we're going to start by looking at the anatomy of skeletal muscle, okay? Now, skeletal muscle is one of the unique tissues of the body. It's one of the only contractile tissues, meaning that it can do something kind of like the heart can or smooth muscle, but it's unresponsive to our voluntary control, okay? And so skeletal muscle is highly specialized in this way. We know that it's innervated by motor neurons, and it is a part of the voluntary or the somatic nervous system, okay? So we're really gonna focus on looking at the anatomy of skeletal muscle. We'll talk about how muscle contraction is brought about and how muscle can generate force to overcome loads, right? To lift things, to move the body. And so as we speak about the anatomy and the function of skeletal muscle, it's really important for you to associate this tissue with movement, okay? Very highly specialized tissue for movement. Let's talk a little bit about what muscle looks like on gross inspection to the naked eye. So it's connected to 2 or more bones by a tendon. We saw a few tendons in the labs. And so a tendon is the non-contractile part that attaches a muscle to 2 bones, or to a bone at 2 points. Okay, now if you look at muscle closely, if we took one muscle, say the biceps muscle here, and we looked at it with our naked eye, we would see it wrapped in this connective tissue sheath that's called the epimysium. So the epimysium is this outer connective tissue layer. It's a bit transparent, and you can see it on most muscles. And it sort of compartmentalizes the biceps from the triceps, from the other muscle groups in the area. Now, if we were to unwrap the epimysium and look a little bit closer, we'd see a bunch of these grouped fibers that are called muscle fascicles. And so within the muscle itself, there are these individualized groups called muscle fascicles, and they are separated by the perimysium, right? So the perimysium wraps around each of these individual fascicles. Now, if we go even further, within a fascicle, there are about 100 to 1,000 muscle fibers. So again, looking within one fascicle, we'll see about 100 to 1,000 of this structure, which is the structural and functional unit of muscle, the muscle fiber. That's synonymous with the muscle cell. So a muscle fiber is the same thing as a muscle cell. Okay, and that's the single structural unit of muscle. And that is wrapped in the endomysium. So it's another connective tissue layer that singles out one muscle fiber from the muscle fascicle. So let's go over that again. We had a muscle fiber wrapped in endomysium. Many muscle fibers make a muscle fascicle. A muscle fascicle is wrapped in perimysium. Many muscle fascicles make a muscle, and a muscle is wrapped in epimysium, okay? Now, if you look even further, there are still smaller subunits within a single muscle fiber. There are myofibrils. Myofibrils are these structural units, these contractile components that help the muscle to shorten. And we'll see exactly how myofibrils bring about muscle contraction. So the magic really happens down at the level of the myofibril. This is within a muscle cell, so highly microscopic. This is not something that we can see with the naked eye, okay? Some features of skeletal muscle: it is multinucleated. So within a single muscle fiber, we have many nuclei. That is not the case with most of our eukaryotic cells. They usually have one nucleus. It is covered in a plasma membrane that's uniquely called the sarcolemma, okay? So the plasma membrane of our typical cell is called the sarcolemma in the case of muscle. And then the cytoplasm, which is that fluid within the cell that helps to nourish and provide all the nutrients the cell needs, has all the organelles for cellular functioning. This is called the sarcoplasm in the case of muscle. So there are a few unique names to skeletal muscle, sarcolemma being the plasma membrane and sarcoplasm being the cytoplasm. Okay, so we spoke about what a muscle looks like, right? We talked about the tendon attaching to the bone, the entire muscle belly, the fascicle, the fiber. At the level of the myofibril, there are a few protein filaments that are moving with each other to bring about muscle contraction, okay? So basically it is down to this level, it's down to these protein structures that are moving to move the muscle, to move the myofibril, to move the muscle fiber, to move the fascicle, and then to move the entire muscle. Okay, so I really want you to get that. The filamentous part, the myofibril part of the muscle is where the functionality of muscle fiber is. That's the area that's actually moving, that's shortening, okay? All right. Any questions so far? Any questions so far? Okay, let's continue on to describe the structure of muscle. Now, we're gonna have to really envision here, because again, this is not something we can see with the naked eye. What we do see when you look at muscle with the naked eye are these striations, and the striations are the myofibrils and the muscle fibers, okay? And so if we were to take a microscope from here on and look even closer, we will see some of the organelle structures that are within a single muscle cell. So again, structures that we cannot see just by looking at muscle. So first of all, there are many myofibrils within a muscle fiber. The muscle fiber is covered or lined with that sarcolemma, which we talked about. So this is the sarcolemma wrapping around this muscle fiber, synonymous with the plasma membrane. Within the muscle fiber, are a few of the organelles that we see in a typical cell. So we typically see the endoplasmic reticulum. In muscle, it's called the sarcoplasmic reticulum. Okay, don't want you to get confused with these structures. They behave very similar to the way they do in typical cells. They're just named differently, okay? So the sarcoplasmic reticulum is synonymous with the smooth ER of a typical cell. Within muscle, we see many mitochondria. Why do you think that is? Why do we have many mitochondria in muscle? Why do you have many mitochondria? More energy, good. So the release of ATP, oxidative phosphorylation, all that good stuff, which generates energy to actually move this machinery. So lots of mitochondria. There's also these transverse T-tubules. So the transverse T-tubules are these green structures, and these are like tunnels or invaginations that run deep throughout the muscle. So this is a horizontal or cut surface view. And you can see that these invaginations run deep throughout the muscle, these T-tubules. And the function of these T-tubules is to propagate that end plate potential. Okay, so we talked about how an end plate potential is generated on muscle at the neuromuscular junction. And once we excite that motor end plate, we need to propagate that voltage change throughout the muscle very quickly. This helps the entire muscle to contract synonymously, okay? So having these tunnels or these invaginations that run deep into the muscle. So on the surface, we're just seeing this area, but understand this goes right through the cut surface of the muscle, and that helps to propagate that membrane potential change, that end plate potential throughout the entire muscle. Now, there are also these terminal cisternae, and the terminal cisternae or lateral sacs are these areas of the sarcoplasmic reticulum that are adjacent to the T-tubules. All right, so understand that the sarcoplasmic reticulum, which is this blue structure here, is doing just what it does, again, in a traditional cell. It's storing calcium, and so having it closely related to the T-tubules is going to be really important with coordinating that membrane potential change and calcium release from the sarcoplasmic reticulum. Very important coordination. So on either side of the T-tubules, we have these lateral sacs, which are basically storage areas for calcium. And together, these three structures form what's called a triad. So a triad is made of a T-tubule, that invagination in the middle, and these two sacs at the end that are storing lots of calcium to be released upon that motor end plate potential. All right. Now, when we look closer at the myofibril, so looking again closely at this structure, we see that this is what gives skeletal and cardiac muscle its striated appearance. This is what makes skeletal and cardiac muscle appear striated, right? That string-like or rope-like texture that we can see with the naked eye is brought about by the striations of these structures. And we don't see this with smooth muscle because there aren't any myofibrils, okay? Very important connection that we'll make later on as well. There aren't any myofibrils in smooth muscle, and that's why we call it smooth. It has no striations, right? Just cardiac and skeletal. Now there's the arrangement of these two proteins, these two filamentous proteins, actin and myosin. And again, looking closer at the myofibril, we can see two different staining patterns here. In the blue is actin, and in the red is myosin. And the arrangement of these two proteins is what forms the myofibril, okay? Now we call the actin filament the thin filament, and we call the myosin filament the thick filament, okay? That's just because of their staining pattern, which we will get into in a minute. So we got actin on the side here in blue, and then myosin in the middle here in red. And actin is the thin filament because it's thinner. Myosin is the thick filament because it is thicker. And these are running in repeated units across the length of the myofibril. These form what are called sarcomeres, okay? Sarcomeres. So I'll try to repeat all that again so it makes a little bit more sense. We have a muscle fiber, right? So this is our muscle fiber. Within our muscle fiber, we have thousands of myofibrils. These are filamentous protein units. On a single myofibril, we have these repeating units that are called sarcomeres. And if you look closely at one sarcomere, so from this structure to this structure, this is a sarcomere. This is made of these two proteins that we just described. In the middle is this thicker protein that's called myosin. It forms the thick filament of this sarcomere. And towards the ends of the sarcomere are these blue proteins, or these thin filaments, which are formed by actin. And together, the actin and myosin overlap in this unit to form